All right, so this is going to be the Lab 8 review, which is going to cover the basal ganglia and cerebellum. We're going to start with the cerebellum and then head on into the basal ganglia. So, brief mission statement. You've heard this before, but Scrubs is a student-driven initiative that is aiming to develop supplemental resources for current and future cohorts that will pass through Brody, and we work on a variety of sub-teams to help offer a unique perspective to students, as we are students who have walked through these courses ourselves. We're hoping that this will become a staple of the Brody student body, and if this is something that you are interested in, we are always looking for more people to join the team. And as always, a brief disclaimer that this is a supplemental aid to your course material. This is not definitive in any way, and it should not, if there are any contradictions between this material and the instruction that you're receiving from the Brody faculty, please go by your instructor's material, because material can be updated over the years, or we can have errors in this resource, although we do our best to limit those as much as possible. And with that, let's go ahead and get into some of the identification of the cerebellum. Now, in the course, you're going to talk a lot about the different pathways of the cerebellum, the different pathways of the basal ganglia. The purpose of this review is mostly to focus on structural identification. And then from there, you can go through the course material and cross-link structure with the function. But for now, we're just going to mostly focus on the function. So start with folia and fissures. So the folia and fissures of the cerebellum roughly correspond to gyri and sulci of the cerebrum. So the folia are going to be these outpouches, okay, which corresponds to a gyrus, for instance. The fissures are going to be in between these. So the fissure is located here. Here is another fissure. You've got fissures located all around. And so these are going to be corresponding to a sulcus for the cerebrum. So think about a folia and a fissure similar to the way that you think about a sulcus and a gyrus. Next, you're going to have lobes. So you've got an anterior, posterior, and flacconodule lobe. Let's start in the front. Here is our anterior lobe. Okay, we go from anterior lobe to our posterior lobe down here. Okay, anterior, posterior. These are separated by this fissure located right here. This is going to be your primary fissure. Your primary fissure separates your anterior and posterior lobe. And if you look, this is the one fissure that we can see that's going all the way down to the core of this white matter. All these other fissures don't quite get down to this central core. So that primary fissure separating the anterior and posterior lobe. The posterior lobe is separated from the flacconodular lobe by this fissure located right here. This is the posterior-lateral fissure. So posterior lateral fissure right there, and this structure is your nodule. So this is the vermal portion of the flacconodular lobe. Now this, giving a brief orientation, this is our fourth ventricle. Remember, this is posterior, this is going to be anterior. The roof of the fourth ventricle, right here, has a little bit of cerebellum that's associated. This portion of the cerebellum is going to be your lingula. Okay, so your lingula is associated with the superior portion of the fourth ventricle. Okay, with your superior medullary velum, located right here. Now, I want to go over some of the other specific structures you need to know from this mid-sagittal view of the cerebellum. So we talked about the nodule. The nodule is separated from the posterior lobe by this fissure located right here. That's your posterior lateral fissure. If we go directly inferior to that, this region of the cerebellum here, this is going to be your uvula. Okay, so your uvula is the vermal region located right here in the anterior portion of the posterior lobe. If I go further back, we've got a region that looks kind of like a triangle. This triangle-looking region is called your pyramid. So this is your cerebellar pyramid, or pyramus, depending on what resource you're looking at. Okay, and then the last thing I want to point out is the pyramus is separated from the rest of the posterior cerebellum via this fissure right here. This is going to be your horizontal fissure. Lastly, I want to point out the cerebellar tonsils. The cerebellar tonsils are the more inferior portions of the cerebellum. And these are actually, when you have cerebellar herniation through the foramen magnum, these are the first things that typically herniate and go through the foramen magnum. So it's important to recognize your cerebellar tonsils. Another fun thing is that the cerebellar tonsils sit on either side of the uvula. So the uvula is dangling between the cerebellar tonsils. And we'll see that in a little bit more detail as we move forward throughout the rest of these images. Now, here we're looking at a superior and inferior view of the cerebellum. From a superior view, the main thing that I want to point out is that you can see this middle raised region of the cerebellum. this is going to be the vermal region. Okay, the vermal region located right in the middle. Outside of the vermis, a little bit more laterally, this will be the paravermal zone. So the region just lateral to the vermis. Lastly, the primary fissure is a little bit difficult to see here, but it's going to be this fissure running right here. This primary fissure is separating, again, the anterior lobe from the posterior lobe. Now, if we look from an inferior view, we have a few more structures that we can identify. The first is we're going to look at the vermis, which is running right down the middle. So there goes your vermis. Then I'm going to point out the tonsils, cerebellar tonsils. They are going to be located right here. And then remember, the tonsils are on either side of the uvula, which is going to be in this region. And lastly, we have the nodule. And we can kind of see the wings of the flacconodular lobe going out, but that'll be much easier to see on our next slide. Okay, so when we're looking for an anterior view of the cerebellum, there's a couple of different structures that we can see. But first, I want to point out that we are now looking at the cerebellum after the brainstem has been removed. The brainstem was attached to the cerebellum via these cerebellar peduncles. You had a superior cerebellar peduncle on either side, a middle cerebellar peduncle, and an inferior cerebellar peduncle. And that is what is connecting the cerebellum to the brainstem. When the brainstem is removed, we are now looking into the back of the fourth ventricle. If we're looking into the back of the fourth ventricle, we know we have the superior medullary velum. These pieces of the cerebellum directly inferior or posterior to that would be the lingula. We come anteriorly and we see the nodule. Going down from the nodule, this is going to be our uvula. And on either side of our uvula, we have our tonsils. So tonsils on either side of the uvula, nodule, superior medullary velum. This region right back here, we can say is corresponding to the lingula. We have our superior, middle, inferior cerebellar peduncles. And then lastly, separating the nodule from the uvula, remember we have that posterior lateral fissure that is going to be located right here. And then I want to point out the nodule is the vermal region of the flacconodular lobe is the nodule, but the rest of this comes out like wings. So here goes your entire flacconodular lobe coming out like wings, with the nodule being in the most central region. Okay, now we need to know a few of the nuclei that are in the deep regions of the cerebellum. There's three that you need to know: the dentate, interposed, and fastigial nucleus. The dentate is most lateral. The fastigial nucleus is located most medial. So let's look at this cross section. This is our pons, so we know this is the anterior portion of the brain. Back here we have posterior. The dentate nucleus is relatively straightforward to identify because it's going to be very squiggly. So right here is your dentate nucleus. So there goes your dentate nucleus right there. More medial to that is going to be the interposed nucleus. The interposed nucleus is located right here. So dentate, interposed, and then fastigial is going to be the most medial. So in total, you're going to have 6 nuclei, 3 on each side: dentate, interposed, fastigial. Here they go one more time: dentate, interposed, fastigial. And we're going to see those on another cross section in just a second. So here goes another cross section so we can see these with a different staining pattern. Again, we're going to start with the dentate because the dentate is most lateral. Again, it has a bunch of squigglies. So that's how I always identify. I look for the most squiggly looking structure in the back of the cerebellar region. And here we go. This is going to be the dentate nucleus. We go more medial, and we have our interposed nucleus. We go most medial, and we have our fastigial nucleus. And then the same thing will happen on the other side. And in this particular cross section, you can't really make out the dentate nucleus on this portion of the brain, but we can see it on this side over here. Now, I also want to point out something that you do need to keep in mind when you're looking at this cross-section of the brain. Remember your vestibular nuclei and your central tegmental tract, because those will come up as well. So central tegmental tract is located right here. The central tegmental tract is communicating between your red nucleus superiorly and your inferior olivary nucleus inferiorly. Okay, and that is an ipsilateral communication. So ipsilateral red nucleus above and ipsilateral inferior olivary nucleus below, central tegmental tract. And then the vestibular nuclei in this particular cross-section are going to be your superior and lateral. Okay, so you have a superior and lateral vestibular nuclei located in the vestibular area over here. And again, if you go back to one of our previous videos, you can walk through the process of how we delineate that. But it's important to know that the superior and lateral paired together, and the inferior and medial are paired together on their respective cross-section orientations. Okay, now getting into the cerebellar cortex. The cerebellar cortex has 3 different layers that you need to know: the molecular layer, the Purkinje layer, and the granular layer. And I like this diagram over here in the bottom left because it does a relatively good job of showing you what structures are going to be located in each one. You're going to discuss this more in class, so I'm not going to spend time going over it, but just recognize that there are different components that make up the molecular, Purkinje, and granular layers. But what you need to know for lab is how to identify these on a histological section. So we're going to start up at the top. The molecular layer is the most superficial of all of these layers, and here it is here. So that is your molecular layer. The single layer at the bottom of the molecular layer is going to be your Purkinje cell layer. Okay, so it's a very small layer located right here. Then you see this darkly stained region. This is going to be your granular cell layer. Okay, so your granular cells are all of this darkly stained region here, and then in the middle of the granular layer, you're going to then have white matter, and that is going to be your communication fibers. So one more time: molecular, Purkinje, granular. And some people remember this as miles per gallon. So MPG, going from superficial to deep. Now we're going to move on into some of the deep brain regions. Here we're going to be talking about the basal ganglia and some of the white matter tracts. So we're going to start with the caudate nucleus. The caudate nucleus has a body and it has a tail. So the body of the caudate nucleus is forming the lateral wall of the lateral ventricle. So here is the body of the caudate nucleus. The caudate nucleus also has a tail. The tail of the caudate nucleus is going to be the roof of the temporal horns of the lateral ventricles. Not great to see in this image. We'll see it better in an image coming up, but that would be located right here because this is our temporal horn, and the roof of the temporal horn would be the tail of the caudate nucleus. Now, when I start going through the deep brain regions, one of the first things I look for is the putamen. The putamen is going to be located right here. Okay, so there goes the putamen. It's nicely delineated. I then go from the putamen to the globus pallidus externa to the globus pallidus interna. So the major structures you need to know at this point is the caudate nucleus, putamen, globus pallidus externa, globus pallidus interna. And then in the center we have our thalamus. Now there is one more gray matter region that you do need to know at this point, and that is going to be the claustrum. The claustrum is a thin slip of gray matter that is going to be lateral to the putamen. So here's our putamen. You go lateral, and you see right here we have the claustrum. So if you recognize where the putamen is, this is one of my main orientation markers. I go lateral to that, and this strip of gray matter will be our claustrum. Now, that is the gray matter that you need to know. We also need to know the white matter that separates each one of these gray matter structures. So we're going to start with the internal capsule. The internal capsule is cutting through right here. Okay, so there goes your internal capsule. You can so think about the putamen and the globus pallidus interna and externa grouped together as a single structure. The internal capsule is going to split that from the thalamus. So going right down the middle. Then if we look external to the putamen, so on the outside of putamen, this thin strip of white matter is going to be the external capsule. So external capsule located right here. And then if we go to the claustrum and go outside of the claustrum, you see that there's a little bit of white matter separating the gyruses from the claustrum. That is going to be your extreme capsule. So it goes internal, external, extreme capsule located most laterally. Lastly, we need to delineate the lateral medullary lamina from the medial medullary lamina. Now, these medullary lamina are going to be separating the structure, the putamen, globus pallidus externa, and globus pallidus interna from one another. So, the lateral medullary lamina separates the putamen from the globus pallidus externa. Okay, so it is this thin slip of white matter right here. And then the medial medullary lamina is going to separate the globus pallidus externa from the globus pallidus interna. And it's a little bit difficult to see here, but it's going to be this strip of white matter right there. Okay, so going through these all one more time really briefly, because these are definitely going to be tested. This is the body of the caudate nucleus, putamen, globus pallidus externa, globus pallidus interna, claustrum. So those are your gray matter areas. Then your white matter, internal capsule, external capsule, extreme capsule. Then you have your lateral medullary lamina, and your medial medullary lamina. And that should take you through a general overview of the gray and white matter from this particular cross-section. Now, again, it is important to know the different regions of the white matter because these can be tested. So we're going to go over this in this particular cross-section, which I think does a good job. Let's start with the caudate nucleus. The caudate nucleus, again, is the lateral wall of your lateral ventricle. So here goes your lateral ventricle, and that is going to make this your caudate nucleus. Okay, so here goes your caudate nucleus right here. Over here, you have your lenticuliform nucleus. Your lenticuliform nucleus is the putamen plus your globus pallidus interna and externa. So we can't delineate the interna and externa here, but we can see the putamen and would be located in this region. So this is going to be the lenticuliform nucleus. Now, if you look lateral to the lenticuliform nucleus, you see a white matter area that is going to be your external capsule. Then you see a thin sliver of gray matter. That thin sliver of gray matter is going to be the claustrum. So here we're just looking at a different view of a cross section. So caudate, putamen, claustrum. Okay, then that means that this has to be our internal capsule, our external capsule, and our extreme capsule. Now I want to point out that the internal capsule has different regions, and these regions can be tested. So first, you're going to have the anterior region up here. Then as it's splitting between the caudate nucleus and the putamen and making this curve, that is going to be the genu of the internal capsule. So anterior, genu, posterior. Okay, and that's going to be the different components that you're going to be able to identify from this particular cross section. Now, we've been looking at white matter and gray matter from cross sections at a more posterior view, but now let's look at what happens if we move anterior and the thalamus isn't present. First, again, let's identify our lateral ventricles. I know that the lateral wall of the lateral ventricle is the caudate nucleus. So I'm going to identify the caudate nucleus right here. Now, here is a region of our internal capsule. And we know splitting the caudate nucleus and the internal capsule, we come over here and we're going to find the putamen. But this doesn't look like the structures that we've been seeing because the thalamus isn't present. So we're actually seeing that the caudate nucleus and putamen are coming together. And that region where the caudate nucleus and putamen come together is going to be your nucleus accumbens. So nucleus accumbens is located anteriorly. Now, right here would be where you would have your globus pallidus, interna and externa, but at this region, we can't delineate what is interna and what is externa. I do want to point out we can still see the claustrum, this gray matter area over here lateral to the putamen. That means that this is our external capsule and this is our extreme capsule located most laterally. And then again, because we said we are anterior, the thalamus is not present, so we are anterior. We can see that the internal capsule region that we'd have in this region would be the anterior limb of the internal capsule. Okay, as we come towards the end, we've got a few more structures just to go over. I want to point out that in this particular brain region, we still have our lateral ventricles, so that will make it easy to identify the caudate nucleus, which is the lateral wall of the lateral ventricles. We have our putamen, globus pallidus externa, globus pallidus interna, claustrum located most laterally, internal capsule, external capsule, extreme capsule. And then if we wanted to, we could go through and we could delineate the white matter tracts that separate the putamen and globus pallidus externa, and the white matter tract that separates the globus pallidus externa from globus pallidus interna. But there are a couple more structures I want to identify that we haven't been able to see very well on the other cross sections. So let's look at the ansa lenticularis. The ansa is this white matter region right here. So there goes your ansa. Next, your thalamus, again located in the middle. But because we have our thalamus, I want to point out the subthalamic nuclei. The subthalamic nuclei are located right here, just underneath of the thalamus. Now, these aren't present in all of the cross sections in a very clean way, but you do need to know that subthalamic nuclei are located inferior to the thalamus, because these can come up and are tested. Okay, lastly, for our last cross section, I want to point out some of the same structures we've been seeing, but in a different view. So here goes our caudate nucleus. This is the body. I want to point out now we can get a little bit better view of the tail of the caudate nucleus. The tail of the caudate nucleus, again, is the roof of the temporal horn of the lateral ventricle, which is located right here. So the roof of that is going to be the tail of the caudate nucleus. The putamen is still, we are able to see that over here on both sides. The globus pallidus intern and extern are a little bit difficult to see, but we can still see the claustrum. So that means we can still see the internal capsule, external capsule, extreme capsule. I do want to point out in this particular cross section, we can see the red nuclei. So the red nuclei here, remember those are part of the midbrain. So there is a region of the midbrain you can see on a coronal cross section. So there are the red nuclei, and then lateral to that, you have your subthalamic nuclei. So red nucleus, subthalamic nucleus. And right here is your substantia nigra. Okay, so there are, again, regions of the midbrain that you can see on a coronal cross section. That is an important point to know because you will most likely get tested on an image with this particular cross section. Okay, I think that brings us to an end of all of the gross identification, but I do want to show you a couple of helpful study images that I've come across in my studies, and I'm just going to flip through these, spend about 5 seconds per slide, just so that way you have access to these as you are studying. And with that, that'll bring us to the end of this video.